Hello friends, in this video we are going to discuss about bacterial conjugation in detail. So what is bacterial conjugation? It is transfer of genetic material from one bacteria to other bacteria by direct contact between the two. So in bacterial conjugation, two bacterial cells come in direct contact with each other. It was discovered by Joshua Lederberg and Edward Tatum in 1946. Bernard Davis in 1950 provided the evidence for cell-to-cell contact through his U-tube experiment. Some of the important points related to conjugation are all or part of donor genome is transferred. So in conjugation, all or part of the donor genome is transferred from one bacterial cell to the other. For conjugation, physical contact is required between two bacterial cells. Conjugation involves different types of plasmids. Conjugation involving F plasmid is most common. Bacterial cells which have F plasmid, also known as sex plasmid, are known as donor male or F plus cells. And those without F plasmid are known as recipient female or F minus cell. F factor exists in two different states. Autonomous state, in which F plasmid is autonomous to the host chromosome and replicates independently of host chromosome. Second is integrated state, in which F plasmid is integrated in host chromosome and hence replicates along with host chromosome. So F factor or F plasmid is known as episome. This we have already discussed in types of plasmid video. You can refer my video, the link is given in the description box. Now let us see some of the bacterial cells on the basis of F plasmid. So this is bacterial cell in which only bacterial DNA or bacterial chromosome is present, but F plasmid is not present. This type of the bacterial cell is known as F minus cell, female or the recipient cell. In this, bacterial DNA is present and F plasmid is also present in autonomous state. This is known as F plus cell, male or the donor cell. In some cases, this F plasmid is integrated in the bacterial DNA or bacterial chromosome by site-specific recombination. Then, this type of the bacterial cell is known as HFR cell or high-frequency recombinant. So, based on this property, that means F plasmid is whether autonomous or integrated. Conjugation is of three types. First is F plus F minus conjugation. Donor cell which contain F factor in autonomous state is known as F plus cell, and the conjugation between F plus cell and F minus cell. F minus cell in which F factor or F plasmid is not present leads to transfer of only autonomous F factor. Now let us see the role of F factor in conjugation. F plasmid houses cluster of 25 that is transfer genes. It determines the expression of sex pili, synthesis and transfer of DNA during mating, and other functions. Now the mechanism or steps involved in F plus F minus conjugation. F plus bacterium with 1 to 3 sex pili bind to specific outer membrane protein on recipient bacteria. Then an intercellular cytoplasmic bridge is formed between F plus and F minus cells. After that, endonucleolytic nick occurs in one strand of F plasmid at origin. After that, 5' end of nicked strand is transferred through conjugation tube into recipient cell. So the direction of transfer of the strand of F plasmid is 5' to 3'. This is coupled with rolling circle replication of the intact circular strand in the donor cell and displaced strand in the recipient cell, as F plasmid is double-stranded. So after conjugation, both bacteria are F plus cell, as F plasmid is now present in both the bacterial cell. and these are present in autonomous state. Now let us understand conjugation between F plus and F minus cell with the help of this diagram. So this is the bacterial cell with F plasmid. This is chromosomal DNA, this is F plasmid, and this is sex pilus. It is known as F plus cell or donor cell. Here the F plasmid is in autonomous state. This is bacterial cell without F plasmid. It has only bacterial DNA. It is known as F minus cell, recipient cell or female cell. Donor cell, with the help of a sex pilus, attaches to the protein present on the outer membrane of recipient cell and pull the recipient cell close. Then cytoplasmic bridge is formed between the donor and the recipient cell. After that, nick occurs in one strand of the F plasmid at origin. Then this nick strand is transferred from donor to recipient cell in 5-3- direction. So now the donor cell has one intact strand of F plasmid. Then complementary strand of this intact strand is synthesized in the donor cell. Similarly, complementary strand of the displaced strand is synthesized in the recipient cell. So at the end, both the cells have F plasmid in the free state. So both are known as F plus cell. So this is the original donor cell and the recipient is now new donor cell. Now second is HFR F minus conjugation. Sometimes F plasmid is integrated into the bacterial chromosome, which we have already discussed earlier. And this integration is mediated by IS elements by site-specific recombination. Such cells are called HFR cells or high frequency recombination strains because donor can transfer chromosomal genes to recipient with high efficiency and hence recombinants are formed with high efficiency. This transfer begins from the origin within F plasmid and the mechanisms or steps are same as in F plus F minus conjugation, but there are certain differences. Portion of F factor which is integrated is transferred first and this transfer is followed by sequential transfer of chromosomal genes. Therefore, frequency of recombination is high or the frequency of formation of recombinants is high. And once the chromosomal genes are transferred, then remaining part of F factor is transferred last. This remaining part of F factor contains ragine which determines the expression of sex pilus and other functions during conjugation. But this entire process takes long time. Hence, in most cases, sex pilus and conjugation tube breaks before the entire chromosome is transferred. As a result, entire set of F plasmid is not inherited by F minus cell and recipient cell are phenotypically F minus. This is because 5' end of F plasmid enters first, but portion at the 3' end enters only after entire chromosomal DNA enters into the recipient cell. And since the conjugation tube and chromosome breaks before entire F plasmid is transferred and hence these are phenotypically F minus. In the rare cases where the entire HFR chromosome is transferred, then the recipient cell becomes an HFR after conjugation. Now let us understand conjugation between HFR cell and F minus cell. So this is HFR cell which is formed when F plasmid is integrated in the bacterial chromosome or bacterial DNA by the site-specific recombination. This is sex pilus and this is F minus cell in which F plasmid is not present. Then nick occurs at the origin, present in one strand of the integrated F plasmid, and then this nicked strand is transferred from HFR cell to F minus cell in the 5' to 3' direction. So here, first of all, portion of F plasmid is transferred, then chromosomal DNA is transferred, and then the remaining region of F plasmid is transferred. So what we see that it has high efficiency of formation of recombinants, as the bacterial chromosome is transferred from this bacterial cell to this bacterial cell with high efficiency. So there is high efficiency for the formation of recombination or recombinant cell. This entire process of transfer takes long time. So before the entire strand is transferred, cytoplasmic bridge breaks. Now what happens? Only portion of F plasmid and portion of bacterial chromosome is transferred. As a result, the cell which we get is still F minus, because entire strand of F plasmid is not present. This region, which is the TRA region or which has TRA genes, is not transferred. This TRA gene is responsible for the expression of sex pilus and other functions of the donor cell. And since in this cell it is not present, and hence it does not function as the donor cell. And this cell is HFR cell. Third is F prime F minus conjugation. Bacteria containing F factor and a part of chromosomal DNA integrated in it is known as F prime bacteria. This results due to excision of F plasmid in HFR strains along with other genes on the bacterial chromosome located at some distance from integration site. So we know that in HFR strains, F plasmid is integrated in the host chromosome at the integration site due to some induction. If this F plasmid is excised along with other genes on the bacterial chromosome, then it results in F prime bacteria. In the F prime cell and F minus cell conjugation, frequency of recombination is high. Frequency of transfer of whole F factor is also high. Hence, after conjugation, both bacteria are F prime cell. Now let us see conjugation between F prime and F minus cell. So first of all, let us understand what is F prime cell. This is HFR cell in which F plasmid, which is shown by red color, is integrated in the bacterial DNA, which is shown by green color, by the site-specific recombination. Sometimes what happens due to certain induction, like presence of ultraviolet radiation or certain chemical, this F plasmid is excised from the chromosomal or from the bacterial DNA or bacterial chromosome with some of the genes of bacterial DNA that is close to the integration site. So now cell which we get has bacterial DNA and F plasmid with some bacterial genes. So this is known as F prime cell. When the conjugation occurs between F prime cell and F minus cell, then nick occurs in one of the strand of this F plasmid. Then this nick strand is transferred from F prime cell to F minus cell in 5' to 3' direction. Entire F plasmid along with some bacterial genes are transferred from F prime to F minus cell. As a result of which, what we get? Original F prime cell and also new F prime cell. In this case, the entire F plasmid along with chromosomal gene is transferred from F prime to F minus cell. So there is high frequency of formation of recombinant. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.